Hello, everyone. This is the first lecture, and to really understand microbiology and understand the bacterial pathogens that we'll talk about at the end of the course, you really have to understand basic chemistry because when I talk about cholera and how it triggers something called rice water diarrhea, it does this through basic chemical interactions with a protein produced by the bacteria and cellular components. So, to begin, all matter is composed of atoms. Now, how atoms interact with one another is basically due to the charge of their nucleus and the electrons. Now, those electrons will interact with various nuclei, allowing to form covalent bonds or weak force interactions. Now, these electrons circle about the nucleus. Electrons are negatively charged. Within the nucleus, there are positively charged protons, and you can remember it P for positive protons, and then neutrally charged neutrons, neutral N, neutral neutrons. Now, these neutrons have mass, and the protons have mass, and the electrons are essentially massless. They're so small. Now, if you take a chemistry course, you'll find out much more information about chemistry. This is just a basic quick review so you have some idea of how it all works. So when we're making covalent bonds, which are strong bonds for biological molecules, you have something like this. We have two atoms here. We have the nuclei, and then you'll see the orange haze around them. Now, this orange haze is where you might find an electron. So in the previous image, we have the two electrons nicely paired in that nice circular form. And that's the easiest way to visualize where they might be. But in actuality, when you look at the actual physics of it and the actual chemistry of it, that orange haze that you see there, that's where you might find an electron at any given nanosecond at any given time. And so when you have these two positively charged protons near each other, surrounded by their electrons, the electrons are attracted to the positive charge. Now, in the basic chemistry, opposites attract. Okay, so that means the electrons, the negatively charged electrons, will be attracted to the positively charged protons. And so as you can see, as these two protons, the nuclei of the atoms, move closer together, the electrons eventually start interacting with both. Instead of simply surrounding a single proton, they now spend part of their time around both. And as you can see, that orange cloud is now surrounding both protons. And now you have a hydrogen molecule. That is a covalent bond. And covalent bonds are the equal sharing of electron pairs. And that's very important. So covalent bonds, the equal sharing of electron pairs. So as you can see here, again, diagrammed with both the electron cloud of what actual physics says is happening and the much more simple to see circle diagram, we again have that cloud of electrons surrounding the protons. As the two protons get closer together, those electrons start interacting with both and you get that covalent bond. And so here you can see electrons are shared in covalent bonds. And one really important fact about this is regular covalent bonds, it's the equal sharing of electron pairs, in that each proton or each nucleus of those two atoms have an equal time with the electrons. The electrons circle one and the other equal amounts. So it's the equal sharing of electron pairs for covalent bonds. And thus you don't have more electrons on one nucleus or fewer electrons on the other, same electrons equally shared between both nuclei of the atoms. Now, that doesn't always happen. So you can have polar covalent bonds. Now, this is the unequal sharing. And as you can see, kind of diagrammed in the space filling model, where you have the large red nucleus of the oxygen and the two smaller gray nuclei of the hydrogens, and the blue dots indicate that the electrons are closer to the oxygen atom's nucleus. And so polar covalent bonds are the unequal sharing of electron pairs, and covalent bonds are the equal sharing of electron pairs. So that's very important to keep distinguished. Now, looking at this slide, there are quite a few different ways to visualize molecules. We have the Bohr model, which is kind of my favorite because it shows where those covalent bonds are. The ball and stick model is also usually kind of nice, but the space filling model often obscures various atoms as you get larger and larger. And so I tend to stick with the Bohr model if I can, or go to the ball and stick, but they're all different ways to visualize the same information. One really important thing about biology and chemistry is that the same chemical formula will have different chemical structures. The same chemical formula, different chemical structures. Very, very important concept. So as you see there, we have pentane and 2-methylbutane up at the top. And so pentane is 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbons. And 2-methylbutane, 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbons. Both are 5 carbon molecules and both look very, very different. And so that's really important. For all biological molecules, every single hydrogen, every single carbon, every single oxygen, everything matters. If it's up, if it's down, if it's right, if it's left, all of that is critically important and really, really shows our cells will interact with one form and will not interact with the other. It is critical. And so, because so many things in biology have exactly the same chemical formula, such as C6, H12O6, a simple 6-carbon sugar. There are lots and lots of different forms of that 6-carbon sugar, and that really matters to biology. And so when we're looking at something like that, where you have lots of molecules with exactly the same formula, but different structures, we start calling them different things. So you have cis and trans isomers. Now, the original meaning of cis and trans was used in biology for hundreds of years at this point. So cis means on the same and trans means opposite. And so as you can see here, the cis molecule is going to have the X group or the functional group or another molecule on the same side of a double covalent bond, whereas the trans one, those two X molecules or functional groups are on opposite sides. And so that's very common, so that you would have a cis isomer, which on the same side, and a trans isomer when they're opposite sides. And then you also have enantiomers. And so this is really shown with hands because the enantiomers, as you can see, again, exactly the same chemical formula with the carbon and oxygen and hydrogen at the top with their red sphere. Then you have the central carbon in black. You have a hydrogen in white, a methane group, CH3 in gray, and then the amino group in blue. And you can see exactly the same, but they're opposite, right? You have the two amino groups in the middle facing each other, the two hydrogens at the end facing out, and that CO2H up at the top, the central carbon, and the methane facing out. So they're opposites of one another. And those are very, very, very different molecules to living cells. And so as you can see here, this is kind of what happens with cells and why chemical structures are so important. So here you can see the asymmetrical carbon atoms, the central carbon atom in the black sphere, the upright gray sphere. And then, as you can see, we have entering a yellow pyramid going into the triangle, a purple cube going into the square, and a red sphere going into the circle. And that one lines up. And so that would be something like a signal molecule binding to a cell receptor. And it works because everything lines up. Pyramid to triangle, sphere to circle, cube to square. But then if you have the same exact chemical formula, but in a slightly different chemical structure, you have, again, but now you have a red sphere going to a circle, so that works. But now you have the purple cube going to a triangle, that doesn't work. And now you have the yellow pyramid going to a square. That also does not work. And that really shows how specific biological molecules are. Slight changes, complete disruption of their function. And so that is another key phrase you guys should learn, is that different chemical structures, different functions. Very important, okay? All you need is a slight change and you have completely different functions. And so how these molecules form is all based on the charge of their nucleus, as well as what their electrons are doing. So when we're talking about biological molecules, we're typically talking about fairly small atoms. And so the third shell of valence electrons, as you see here with sodium and sulfur and phosphorus, are pretty much some of the biggest atoms that biological molecules will work with. And so as you can see here, we have the outer valence shell, which is going to be the outermost layer of electrons. We have the inner valence shell, which is the innermost layer of electrons. And for biology and for this class, we're just going to talk about anthropomorphizing electrons, because it's easiest to say what they want and what atoms want. So for most atoms, what they want is a filled outer shell of electrons. And so the inner valence shell of electrons always has 2 electrons, if possible. The next valence shell will have 8. The next valence shell after that will also have 8. And that's pretty much it for biology. We don't really get much bigger atoms than that. And so for us in biology, the inner valence shell always wants to have 2 electrons, and the next valence shell or shells want to have 8. And that would make a happy atom. And so something like oxygen, which has a positive charge nucleus of 8, has 2 inner valence electrons, so it's happy. Then it has 2, 4, 6 outer valence electrons, but it wants 8. Okay, remember the outer valence shell for biology and for this class want to have 8 electrons. So oxygen will essentially look for 2 more electrons to have an outer valence shell of 8. And as you can see here in the diagram, oxygen and sulfur have 6 electrons in their outer shells and require 2 electrons to achieve stability. And that's really what all atoms for biology want to do. And so that dictates how well they will form covalent bonds, either regular covalent bonds, the equal sharing of electron pairs, or polar covalent bonds, the unequal sharing of electron pairs. Now, functional groups are key concepts, and I will use the terminology as such as the name of the functional groups quite commonly throughout this course. And so while I will never ask you guys to draw the shapes, I do want you guys to have a general idea of what they are. So the hydroxyl group is always going to be oxygen and hydrogen. So it's going to be polar. It's going to have a charge, which we'll get into in just a second. And so hydroxyl groups are functional groups. They're just an OH group attached. We talk about DNA and RNA. We'll go way in depth into hydroxyl groups, and I'll talk about them all the time. Now, both aldehyde and keto groups are often important in sugar molecules. So if you take organic chemistry, you'll talk a whole bunch about the aldehydes and keto groups, although I won't usually mention them much in this class, except when I'm talking a little tiny bit about the polysaccharides or the complex sugars. But what I will focus on are the carboxyl and the amino groups. So in my next video, I'll be talking about proteins, and every single protein ever discovered in anything has the beginning, which is always an amino group, and the end, which is always a carboxyl group. And so I'll be talking about that a lot. Now, amino terminus, or the amino group, is usually going to be NH2, or depending on where it is and what suspension or solution it's in, it might be NH3. And the carboxyl group is usually COOH, but depending on the solution or what it's in, it might be COO. But there'll still be amino groups or carboxyl groups. And I will be talking about those a lot. Another key functional group is the phosphate group. So if you've heard of ATP, the energy unit of the cell, adenosine triphosphate, 3 phosphates linked together, 5' phosphate in DNA and RNA, all very, very important. So phosphate groups will be really important. That covalent bond between the R group and the phosphate, you can break that for a lot of energy. And so that's really important. And then we have the sulfhydryl group. And so this is important in proteins. When we talk about proteins, I'll be talking about something called disulfide bridges, which is a covalent bond between two sulfhydryl groups, and that adds stability. So we've gone over covalent bonds, the equal sharing of electron pair, and that gives very strong bonds. Usually you have to have an enzyme make a covalent bond or break a covalent bond in life. And so an enzyme is a protein that does a job, and often those jobs are making strong covalent bonds or breaking those strong covalent bonds. We'll talk about other types of bonds as well, and most of these are weak force interactions in that they can form on their own without any outside input. So ionic bonds, attraction of opposite charges, hydrogen bonds are very special. They have a very precise definition. Hydrophobic interactions are going to be nonpolar interactions. And then van der Waals interactions are very brief occurrences, and they only occur in cell membranes, which is just kind of this waving charge. It's very weak and very brief, occurs all the time, but it doesn't have much bearing on this class. Again, if you take biochemistry or organic chemistry, you might come into it. So, forming molecules in cells. For living cells, oxygen, nitrogen, and carbon are really the only things that will be easily manipulable in cells. So carbon, oxygen, and nitrogen can form double covalent bonds. So one covalent bond is the equal sharing of an electron pair, 2 electrons. Double covalent bonds, the equal sharing of 4 electrons. Okay, and if you're sharing 4 electrons with the same atom, with another atom, you're going to have a very, very strong structure. Okay, it's not going to have a lot of give. It's not going to break apart very easily. So if you have double bonds, you're really increasing the stability of that molecule. Now, carbon, if we go back here, as you can see, carbon has a positive charge of 6 in its nucleus. It has 2 inner electrons, but it only has 4 outer electrons. Now remember, in biology, atoms want to have 8 outer electrons. Now carbon only has 4 outer electrons. And so what that means is carbon has 4 valences. It wants to make 4 more bonds. And so it can form a wide variety of structures, right? It can have 2 double bonds, it can have 4 single bonds, all different kinds. And this is where we really get structure determines function, because carbon can literally make millions of different structures, and it can have millions of different functions. And so in molecular biology, structure determines function, full stop. If you slightly perturb the structure, you've destroyed the function in molecular biology. Now, in something like physiology or anatomy, that changes because we're talking about large-scale interactions. So the structure of your bones determines function, but what you use that function for, such as running versus swimming versus tennis, that will have an effect on the musculature, and thus the skeletal reforming as bone is reabsorbed and put down over and over to better deal with stress. And so runners will change the structure of their long bones and their legs to better deal with that stress. So in anatomy and physiology, you'll often hear structure determines function and function determines structure. And that is true for the whole organism. But in molecular biology, in microbiology, that is completely not true. Full stop, structure determines function. Slight disruption of the structure destroys the function in molecular biology. It's very, very precise at the cellular level. So structure determining function, as you see here, these are all pure carbon. Pure carbon in flat hexagons, pencil lead. We don't care. We'll use it to write with. If you lose a pencil, you don't care because, hey, it's cheap and easy. But pure carbon in the lattice structure, that's a diamond. And we like diamonds. They're shiny. They're expensive. Even lab-made diamonds are not cheap. And so there you see at the molecular level, structure determines function. A lattice shape of carbon, diamond. Flat planes of carbon, pencil lead. So very, very different structures, very, very different functions at the molecular level. So carbon, okay, will make 4 bonds. Now, when we're talking about carbon, it is a 6 plus, a 6 positive charge nucleus. And oxygen is, as you see back here, oxygen is an 8 plus. So it is a technically larger atom. It has a larger, more positively charged nucleus. Remember, in chemistry, opposites attract. So a more positively charged nucleus is more attractive to negatively charged electrons. And because all the protons clump up in the nucleus itself can have a positive charge of 8. But those electrons, they do not clump up. And so they always will have by themselves a negative charge of 1. So if we're looking at our oxygen atom, the nucleus of the oxygen atom is a plus 8. And so when you have something like the hydrogens, which are teeny tiny little atoms, and their nuclei, as you see back here, is a plus 1, that's not super attractive to electrons, which are also negative 1. So when we have something like oxygen in biology, which for biology is a large molecule, or is a large atom. Oxygen is a large atom in biology. That plus 8 nucleus is very attractive to the minus 1 electrons. And so when you have something that is a relatively large nucleus, like oxygen atoms, electrons will want to spend more time with that oxygen's nucleus. So when you have something like hydrogens, which are a plus 1 nucleus, and electrons, which are minus 1, near the oxygen atom's nucleus, which is a plus 8, those negative electrons will spend more time near the oxygen atom's nucleus. Big positive charge, more attractive to negatively charged electrons. And so you get something called polar covalent bonds. Polar covalent bonds, key word, is the unequal sharing of electron pairs. Covalent bonds, equal sharing of electron pairs. Polar covalent bonds, the unequal sharing of electron pairs. So what you see in water, H2O, is those two hydrogen atoms rarely have the electrons near their nuclei. And the oxygen atom usually has those electrons near its nuclei. And you can see this diagram by the blue dots in the space-filling model. You can see the red oxygen has 2, 4, 6, 8 dots of the electrons near its nucleus, and the gray hydrogens have none. Now, electrons are negatively charged. So, if the negatively charged electrons are spending more time near the oxygen atom's nucleus, that oxygen atom picks up a net negative charge. And the hydrogens, because they rarely have the electrons near their nuclei, pick up a net positive charge. Now, it's not a full plus 1 and it's not a full minus 1, but it is the oxygen atom is partially negatively charged, and the hydrogens are partially positively charged. And that is polarity, an unequal distribution of charge. So when you look at something like this, here you can see, again, the electron pairs are closer to the oxygen atom. That oxygen atom is picking up a net negative charge. And because the electron pairs are further from the hydrogen atom's nuclei, those hydrogen atoms pick up a net positive charge. Okay, and so here you can see, again, a partial negative charge on oxygen and a partial positive charge on both hydrogens. And you have a polar molecule. And that totally changes how everything works. It's why we have life on Earth, because of this unequal sharing of electron pairs, giving water molecules polarity. So, hydrogen bonds. Now, opposites attract. And so if you have a partial positive charge on hydrogen, and then you have a partial negative charge somewhere else, those partial charges, positive and negative, are going to attract one another. And so when you look at hydrogen bonds, I have a very specific definition of hydrogen bonds. So hydrogen bonds for me are defined as a partially positively charged hydrogen is attracted to a negative charge on another molecule, such as water, or on the same molecule if we're talking about a larger molecule. Okay, so for water, they're small molecules. A partial positively charged hydrogen will be attracted to the partial negative charge on the oxygen of a different water molecule. And so here you can see diagrammed two H2O molecules. The oxygen on one, negatively charged, is attracted to the positive charge on the hydrogen. You can see the hydrogen bond is that little red dashed line pulling them together. And water molecules will align so that oxygens are pointed to hydrogens at all times if they can. And so in water, liquid water, it's constantly moving. And so you can make and break these hydrogen bonds constantly. They're weak force interactions. They're kind of drawn to each other, but if that molecule is moving about, it can break it just as easily. So liquid water can get quite close to one another because you have a partial positive charge and a partial negative charge, and they're all moving, so they can get close to each other. So liquid water can be dense. Water molecules can pack tightly together, and they're always forming hydrogen bonds between a partially negatively charged oxygen and a partially positively charged hydrogen on another water molecule. And they're constantly forming, constantly breaking, closely packed together. And so, ideally, water molecules want to line up so that always the partially negatively charged oxygen is facing partially positively charged hydrogens, and partially positively charged hydrogens are always facing a partially negatively charged oxygen. And so as you can see here, each water molecule will form hydrogen bonds with up to 4 neighbors. But because we have liquid water and because we have energy, those water molecules will constantly moving. And so this will only occur if water molecules are not moving and you've frozen them and you go from liquid water to ice. Now, because of those hydrogen bonds, ice floats. Because of those hydrogen bonds that form due to polar covalent bonds, we have life on Earth. Because ice floats, we have life on Earth. It's really amazing. So if you have those polar molecules, those water molecules moving around, and you remove the energy, you slow those molecules moving, what they have to do, or what they want to do, is maximize the potential hydrogen bonds. So to maximize those hydrogen bonds, the only way to align correctly is to form this lattice. And so they force each other apart. In liquid water, because those hydrogen bonds are transient, constantly formed, constantly broken, the liquid water gets very close together. But when you freeze water, it has to maximize the hydrogen bonds. And so each water molecule gets further apart, and you have more space between the water molecules, and thus solid water, ice, is less dense than liquid water, and so ice floats. Now, because ice floats, you have liquid water underneath it. Because you have liquid water underneath the ice, you have life. Okay, and so, for example, if you have sea ice, pack ice out in the Southern Ocean, penguins can still dive under that ice, can still go fishing, can still come up and land on the ice, and the liquid water remains life-giving underneath. You can have ice fishing in which you might have, several feet of frozen ice on the surface of a lake, and yet dig down deep enough, you have liquid water underneath, living fish and everything like that. I'll talk later on about the beginnings of life on Earth, and we went through multiple periods where everything was frozen, and yet life could continue because underneath that frozen ice was liquid water in which life could continue. And so as you can see, the arrangement of the water molecules in ice provides space between the water molecules so that it is less dense. And so because of that maximizing of weak forces, because of the maximizing of hydrogen bonds, that maximizing of a partially negatively charged oxygen wanting to align to a partially positively charged hydrogen, ice is less dense than water and it floats. And so as you see down below, liquid water, hydrogen bonds are continually made and continually broken due to the energy of the water molecules, so they can get close together. And then if you really add heat and you have gaseous water, which is going to be water vapor, usually you don't have hydrogen bonds forming. And so we have solid ice. Water molecules are forced apart, so solid ice, frozen water, is less dense than liquid water. As you're melting ice, water molecules are not moving very fast and they can pack in closely together. And so water becomes denser. The colder it is, it becomes denser until it freezes and then it floats. As you add more energy into water molecules, they'll start moving further apart due to their energy. So warm water is less dense than cold water. And then as you continually add energy, water molecules will move faster and faster and further apart until they boil off as steam and water vapor. So hydrogen bonds, as we've just seen, can form between different molecules. So the partially negatively charged oxygen is attractive to the partially positively charged hydrogen on different molecules between two water molecules. But we also see hydrogen bonds all the time in larger molecules. So that orange blob there is a protein. And proteins are held together in part by hydrogen bonds. The partially positively charged hydrogen is attracted to a partially negatively charged atom in the same molecule. Our DNA is held together only by hydrogen bonds. A's hydrogen bond to T's, G's hydrogen bond to C's, and that is the only thing holding our two strands of DNA together. That just hydrogen bonds. Our final structures of proteins are often controlled by hydrogen bonding. So again, hydrogen bonds are the attraction of a partially positively charged hydrogen to a negative charge on the same molecule or on a different molecule, okay? So when we have something like sea ice forming out in the Antarctic Ocean or in the Arctic Ocean, it's going to form in salt water. Now, when I showed you guys the lattice structure of water, as you can see, that's all simple water molecules. So when pack ice forms out on the ocean, water molecules still want to form this lattice. And so as ice freezes out on the ocean, what's going to happen is salt is forced out of the water crystals. Out of the ice crystals. So you get something called brine veins, in which this is super salty water that remains liquid, even if you have something like this structure where we have pack ice or sea ice out there on the ocean. It's going to be mostly freshwater ice. And then through that freshwater ice will be all these thin brine veins. And this is a really amazing microbial ecosystem that can occur over and over again. As that seawater freezes, it forms pure water ice crystals lined with these brine veins. In those brine veins are hundreds of different species of bacteria, hundreds of different species of single-celled eukaryotic organisms. Some are photosynthetic and some are something called bacteriivores that eat bacteria. And so you have these really amazing little microbial ecosystems in this open water sea ice that forms over and over and over again. And so you get these amazing, amazing interactions. You'll have photosynthetic things utilizing the solar energy, forming sugars from light. You'll have bacteria that eat those photosynthetic things. You'll have eukaryotic organisms that eat the bacteria and photosynthetic organisms. Just absolutely amazing little ecosystems just living in this ice. So other amazing facets about water density is you can have completely separate water column ecosystems. And so here you can see in these three pictures, that's a scuba diver diving down through clear water that is less dense than the murky water below. And you can see that scuba diver is entering that murky water. And there's such a density difference between the clear water and the murky water that it doesn't mix, that there is less dense clear water and then more dense murky water. And there is no mixing because of the difference in density. We often see this in the ocean. And so you have these amazingly stratified water columns where the different densities in water really make these sharp, defined differences in water. And that's all based on water density. And so another really neat part of oceans is that due to the change in continents, the recycling of Earth's crust, the change in the shape of the globe, what you have is something called ancient salt deposits from very ancient oceans that had dried up, left their salt, more new crust was put over it. And so in our modern ocean floor, we have ancient ocean floor and more recent crust deposits. And sometimes what happens is those more recent crust deposits reveal that ancient ocean floor, that ancient salt deposit. And that ancient salt deposit can waft up this high salinity into the surrounding water. And so as you see here, we have something called a hypersaline basin. And this will be a depression in the ocean's floor that has reached those ancient salt deposits. Those ancient salt deposits have dissolved into that localized water, making it incredibly salty, much more salty than the rest of the seawater. Now, because that is such deep water, because there is very little mixing that deep, because of that high amount of salt going in, there's very little oxygen. And so what you have are these basins, which is basically very, very unattractive to most life forms. It's too salty and not enough oxygen for most things. But for some bacteria, this is a great place to live. That super salty water they've evolved to, they can utilize the various chemicals in it for energy, and they don't need oxygen. And so you get these really unique ecosystems forming in these ancient deep hypersaline anoxic basins. And so you have really unique microbes that really like this environment. And you get kind of like this little oasis in the bottom of the ocean because lots of bacteria can use this for energy. And so you get more and more and more bacteria. Now, because that is so salty, that water is very, very dense and will not mix unless there's a very strong impact. Otherwise, it's going to be like essentially atmosphere and ocean. There is no big mixing. It's not going to dissolve away. It's going to stay as kind of defined layer. And so you can see here in this diagram, there's that interface and the beach. And so that beach area is where the microbes that are using the super salty chemical rich water for food will kind of diffuse out. And when you're talking about the ocean floor, it's like a desert. There's not a lot of life out there. So when you find something where you have lots and lots of bacteria, that's a food source. And so these deep basins actually become these little oases where filter feeders like mussels will line that beach area, filter feeding and eating the bacteria that kind of float towards the surface of that deep basin. And if you have mussels, then you have things that can eat the mussels. And so you'll get crabs and some other shellfish. You might get octopi coming in, cuttlefish, something like that to eat the mussels. Then things that eat small fish or crabs. You'll get things like krill and other crabs that will come eat the dead animals. And so you get this really amazing little oasis just out in the middle of the sea floor due to this water density difference with this super salty water. And you can see in some of these pictures here, again, that murky water lined by mussels, a nice little oasis, totally due to the polarity of water. Okay, so part of hypersaline is having extra salt. And so salt forms something called ionic bonds. So when we're looking at sodium and chlorine, sodium has a plus 11 nucleus, and chlorine has a plus 17 nucleus. So if you're a sodium atom, you have two inner electrons, and that's good. Then your next valence shell has 8 electrons, and that's good. But your outer valence shell has 1 electron. You don't like that. The atom wants, for biology, the atom wants 8 outer electrons. And if you're sodium, you have 1. And there's no way you're going to make up those 7 bonds. You just have this tiny little nucleus, just plus 11. That's not super attractive to 7 more electrons. So sodium typically just kicks out that last electron. And so sodium becomes a sodium ion. It gets a +1 charge. Now, chlorine, having those 17 plus charge in its nucleus, has 2 inner electrons. The next shell has 8 electrons, which is great. And then the outer shell has 7 electrons. And so all chlorine needs is just that 1 more electron. And so if sodium has kicked out that electron, chlorine is very easily takes that last electron, and then it gets 8. And then it is a very happy atom. It has 8 outer electrons. So sodium lost 1 electron and became a +1 ion. And chlorine took that 1 electron from sodium and became a negatively charged ion, a chloride ion. And so sodium is a +1 and chloride is a -1. +1, -1, opposites attract. We get sodium chloride, you get table salt. And so ionic bonds really easily interact with water because water has a partial positive and a partial negative, and salt has a positive and a negative charge. So when you mix those together, the salt crystals, which have a line, so positives touch negatives and negatives touch positives, as you can see here, the negative chloride ion in green and the positive sodium ion in blue, form that salt crystal. Then as you add it to water, those water molecules will surround each and every single ion so that the negatively charged oxygens are going to be facing the positively charged sodium, and the positively charged hydrogens are going to be facing the negatively charged chloride. And as you can see, they're going to form little spheres. And so you see it here as a flat circle, but it is actually a sphere of water molecules surrounding each and every sodium or chloride ion. And that's how salt dissolves in water. And you can see that the more salt you add, the denser packed in those water molecules will be. So water that has a lot of salt dissolved in it is more dense or is denser than water that does not have a lot of salt mixed in. And water will actually pull away anything that has charges. So when you mix molecules in, and let's say this is something like sugar, a sugar crystal will also be dissolved because you have hydrogen bonding. Water molecules will actively pull away anything that has a charge, positive to negative and negative to positive. And so here's another view of the salt crystal dissolving into water. And again, you can see those partially negatively charged oxygens will face in towards positively charged sodiums, and the partially positively charged hydrogens will face in towards that negatively charged chloride ion. Okay, so we went through covalent bonds, regular covalent bonds, the equal sharing of electron pairs, and polar covalent bonds, the unequal sharing of electron pairs. Ionic bonds is the positive charge attracted to a negative charge. Hydrogen bonds, the attraction of a partially positively charged hydrogen to a negative charge on a different molecule or in the same molecule. And ionic bonds and hydrogen bonds are what we call polar interactions or hydrophilic interactions. And in this case, hydro means water and philic means loving. And so ionic bonds and hydrogen bonds are water-loving bonds. Now, another term for hydrophilic is going to be polar interactions due to the polar covalent bonds that make them up, or also charged interactions, because if you're polar, you have a charge, and thus you are also hydrophilic. But let's say you don't have a charge. If there is no charge, then it is water-fearing or hydro-water-phobic interfering. And so hydrophobic interactions are going to be nonpolar interactions where you have normal covalent bonds that have no charge. So hydrophilic is charged, is polar, and hydrophobic is uncharged and is nonpolar. And so water, a polar molecule made up of polar covalent bonds. But ethane is a nonpolar molecule. Now, one of my favorite lines is sharing is caring, and carbon believes in sharing is caring. Carbon makes normal, regular covalent bonds because oxygen is that larger nucleus. It's more negatively charged, more positively charged, excuse me. And so electrons want to spend more time with it. But carbon has a smaller positively charged nucleus. And so it's willing to make equal covalent bonds. And you have equal sharing of the electron pairs. So hydrophilic molecules will have some charge on them. Maybe it's a +1, maybe it's a partial negative. And then those partial positives and partial negatives and full positives and full negatives will want to interact with each other because opposites attract. But if you don't have a charge, you don't want to be with something that has a charge. So anything that is hydrophobic, anything that has no charge to it will clump up, and anything that has a charge will be pushed away. Water will stay away from something that's hydrophobic. Ions will stay away from something that is hydrophobic. So hydrophilic, water molecules, water-loving. Water molecules will surround ions, will surround larger molecules and dissolve it away. But hydrophobic, water stays away. And so something like olive oil in water, olive oil will clump up and stay bound to itself and exclude water. And so hydrophobic molecules will clump together and separate themselves. Now, this allows lots of clever insects to walk on water, where the polarity of water molecules means they're attracted to each other and cause surface tension. If you're an insect, you can have hydrophobic molecules lining your feet, and you're light enough not to disturb the surface tension of the water. So their hydrophobic feet allow them to walk across the surface of the water. Their hydrophobic feet repel the water, and the water retains surface tension. And so here you can see with the green lines, a hydrophilic surface, water-loving surface. Fluid wants to spend the most time with it, so you get a concave structure forming. But if you have a hydrophobic surface, it repels water, and water wants to touch it least, and so you get a convex structure. And then down at the bottom, you can see you have a hydrophobic coating, okay, in which you have water molecules that want to stay far away until you get really defined hydrops. And then if you have a hydrophilic coating, again, water wants to be near it the most, and so you get very flat drops. And so when you use something like Rain-X on your windshield, it's going to be a hydrophobic coating. It wants to make those water molecules and water droplets be away from it, so you get those really tall, defined drops. Now, you can have things that are hydrophilic, in which you have polar charges, and that molecule has, maybe it's a +1, maybe it's a -1, maybe it's a partial positive, maybe it's partial negative. You can have hydrophobic molecules in which there is no charge. And then you can have one molecule that is both. And these are amphipathic molecules. One molecule that is both hydrophilic and hydrophobic. Very important part of the definition. One molecule that is both charged and uncharged. One molecule that is both polar and nonpolar. One molecule that is hydrophilic and uncharged. One molecule that is water-loving and nonpolar. Okay, so it's really important to keep those definitions straight. And so amphipathic molecules are something like soap. You have that zigzag, okay, for a hydrocarbon chain. And in this case, hydro means hydrogen and not water. So it's a chain of carbon atoms and hydrogen atoms. And because carbon believes sharing is caring, that hydrocarbon chain is going to be this case, water, phobic, water fearing. Okay, so hydro can mean hydrogen and hydro can mean water, even when you're talking about the same molecule. So a hydrocarbon tail is a hydrophobic molecule. Hydrocarbons, hydrogen and carbon, hydrophobic, water fearing. Now, the water loving or hydrophilic part will be the head group and is going to be charged. And as you can see, there is a sodium ion there that has a positive charge. And so that is going to be attractive to anything that is hydrophilic or charged. And so therefore, when you mix soap with water, the hydrocarbon tails that are hydrophobic will interact with things like oils and greases, which are also hydrophobic. And the head groups, which are hydrophilic, will interact with the water. And so you get something like this. Soap. Hydrophilic heads want to interact with water, and the hydrophobic tails want to stay away from water, so soap makes soap bubbles. Hydrophilic heads facing in, trapping a small layer of water, and hydrophobic tails facing out. And one neat physics aside is that's why we get that rainbow sheen. The distance between the soap molecules is going to vary. And as the soap molecules get closer and further apart and that soap film changes, you'll have different refractions of the light and you get that rainbow sheen as the light changes refraction, breaking into the rainbow because of the difference between those two soap molecules. That's just a fun physics aside. Now, why do we care about things other than soap? Well, our cell membranes are made out of amphipathic molecules. And so when you have something called a phospholipid bilayer or a phospholipid, it is an amphipathic molecule. And so here you see something called a micelle, a liposome, and then the bottom one is something that looks like a cell membrane. And so a phospholipid, PHOSPHOLIPD, a phospholipid, is that little kind of ping pong ball with the little legs. That is one phospholipid. And so the phospho part, the white part, is going to be hydrophilic. And the two little legs are hydrocarbon legs, and those are hydrophobic. And so hydrophobic legs of phospholipids will always face towards each other, and the hydrophilic heads will face out. And so if you look at the micelle, you can see that you have the ping pong ball white heads facing out. And that would work in blood or sera. It is going to be hydrophilic. And so if you are trying to get a drug to a patient, you can put a hydrophobic drug in the micelle because the hydrophobic tails are facing in. A hydrophobic drug will go into the micelle, and then you can target it to your cell target with the hydrophilic heads facing out. But let's say you have a hydrophilic drug. Well, then you can use the liposome, in which you have a little pocket inside of hydrophilic heads facing in. Your hydrophilic drug goes in there. It is separated from the outside by that hydrophobic tail line, and then you have that outer layer of hydrophilic heads able to target it to a cell. And so this gives us our cell membranes in that we have hydrophilic head groups facing out, a hydrophobic tail group, and a hydrophilic head group facing in. You get separation of the outside environment from the cellular environment. So soap bubbles, hydrophilic heads face into water, and hydrophobic tails face out. But for lipid vesicles, for micelles, for liposomes, for cell membranes, you have hydrophilic heads facing out and hydrophobic tails facing in. And here you can see water on the outside, so you could have extracellular water and intracellular water, and you get cell membranes. So talking about what makes up cells. Typically, living tissues are going to be about 70% water. And this does vary somewhat. Cells that are evolved to deal with very dry environments might have less water. Cells that deal with aqueous environments might have slightly more water. But in general, on average, living tissues are 70% water. And that gives about 30% left over to be other stuff. And again, it varies by cell, it varies by tissue, but then you might have anywhere from 7 to 10, maybe as low as 5 of ions, such as sodium and chloride, cobalt, zinc, copper, iron, as small molecules. And then macromolecules make up the rest, usually about 25%. And these macromolecules are the main building blocks of cells. Now, the majority of macromolecules are proteins, which can be called polypeptides. And so POLY means a lot or many, MANY. So polypeptides have a lot of peptide bonds. Then you have nucleic acids, which will be DNA and RNA. You'll have carbohydrates, which are known as polysaccharides. And saccharide means sugar. And again, poly means a lot. So carbohydrates are a lot of sugar molecules. And then lipids are fat. So when we're talking about this, each one has different functions because structure determines function in molecular biology. So nucleic acids are the genetic material. We have DNA being long-term genetic storage and RNA the expression of those genes. Proteins, a huge number of functions. They can be support, they can be protection, they can be transport, they can be catalysis, they can be enzymes, I mean, all different things. And we'll talk about them in the next lecture. Now, carbohydrates or the polysaccharides are often going to be the source of energy, such as glucose. They'll also be really important carbon backbones or carbon skeletons to make other structures. And then their structural support. Like every single plant you've ever seen is made up of primarily glucose, making cellulose, making their cell walls. I mean, that is a major, major, major structural function of sugar and carbohydrates. And then lipids typically are going to be for energy storage as fats for energy. Then you'll have phospholipids making up cell membranes, and then things like blubber in sea animals to insulate. You'll have steroids and hormones that are going to be lipid-based. Some vitamins are lipid-based. And then all of our axons in our nerves are wrapped around or are wrapped by lipids for insulation. So how do we make macromolecules? Well, joining two monomers, monomer, the basic building block of macromolecules. So joining two monomers together is called condensation, or also known as dehydration. Now, if you think about condensation on your mirror during a shower, it's the same concept. You're condensing or braining out water. And so here you can see two monomers. A hydroxyl group on the first monomer is being joined to a hydrogen on the second monomer. And a hydroxyl, OH, plus a hydrogen makes H2O, and water comes out, and a covalent bond is made between monomer 1 and monomer 2. Then you bring in a third monomer, and so on and so forth. And so you're condensing water out of these monomers, making a macromolecule. And then you have hydrolysis for hydro, water, and lysis, breaking. So hydrolysis, you bring water in to remake that hydroxyl group and that hydrogen and break apart that covalent bond. And so an enzyme is a protein that can do condensation and take water out and make that covalent bond, or a different enzyme, or sometimes the same enzyme, can also do hydrolysis and take in a water molecule and break that covalent bond. And so all the time in our cells, condensation or dehydration reactions are going on, making water, and that water is then used by the cell to do hydrolytic interactions or hydrolysis, breaking covalent bonds apart. So condensation, linking monomers together, making covalent bonds, and hydrolysis, breaking covalent bonds. And so that's always going to be covalent bonds. So to do energy, cells need something called adenosine triphosphate, or ATP. It's the major energy unit of the cell. There are other things like GTP, which can be used as energy, but the main form is ATP. Now you have an adenosine as the main beginning structure of ATP, and you see it as the orange and blue structure there, a ribose sugar and an adenine base linked together, and that's kind of the main structure. Then you can have one phosphate added, monophosphate, MONO means one. You can have two phosphates, diphosphate, DI means two. Or you can have three phosphates, adenosine triphosphate, TRI, meaning three. If you have one phosphate, you cannot use it for energy. You have to build onto that. If you have two phosphates on it, you can kind of sort of use it for energy, but it's not ideal. And if you have three phosphates on it, it is easily used for energy. And so cells typically will use up their entire pool of ATP before they are willing to try and use ADP for energy. It's simply much less efficient. And so the hydrolysis, or the breaking of that covalent bond between the third and second phosphate groups, releases energy. And that energy can be used to make other covalent bonds or break other covalent bonds. Okay, so carbohydrates, our first macromolecule that we're going to go in depth to. So carbohydrates, we tend to think as what we eat, right? Sugars and starches and breads and such. But carbohydrates, also known as polysaccharides, have a lot of other functions. And so carbohydrates as simple sugars, monosaccharides, one sugar, or disaccharides, two sugars, can be something like glucosamine, which is a key and critical component of all of our joints, smoothing the interactions between bones. Polysaccharides, complex sugars, can make up the exoskeletons of things like crabs and insects, and that's the main structure. So when you have something like a lobster's exoskeleton is mostly carbohydrates, mostly complex polysaccharides with some amino acid proteins mixed in. So it really is this incredibly versatile structure because most carbohydrates, most saccharides, have a carbon backbone. And carbon, remember, sharing is caring, can make an infinite number of structures, an infinite number of functions. And as I said earlier, cellulose is simply straight chains of glucose linked together, and it is one of the most common molecules on the earth because all plants have cellulose in their cell walls, giving them structure. So polysaccharides are polymers of sugars, and polysaccharides typically are going to be anything more than about 7 sugars linked together. Oligosaccharides are usually only discussed in something like organic chemistry, so I'll leave that for there, but that's usually between 3 and 7 or 3 and 10 sugars linked together. Disaccharides, the prefix Di means 2, so those are always 2 sugars linked together. And monosaccharides, the prefix mono means 1, and that's always 1 sugar by itself. Now, the number of carbon atoms is how we define saccharides. So a 3-carbon sugar has 3 carbons. A 4-carbon sugar has 4 carbons. A 6-carbon sugar has 6 carbons. And usually, most sugars and most saccharides follow the rule of 1 carbon means 2 hydrogens means 1 oxygen. So a 3-carbon sugar will have 3 carbons, 6 hydrogens, and 3 oxygens. Typically, a 6-carbon sugar will have 6 carbons, 12 hydrogens, and 6 oxygens. But there is a huge variety in sugars. And so here you can see glyceraldehyde at the top, a simple 3-carbon sugar. Then we have 2 5-carbon sugars, ribose and deoxyribose, which have almost exactly the same chemical formula and a very similar chemical structure. And yet ribose has the hydroxyl group on the 2nd carbon, and ribose is present in RNA. And D, the prefix DE means no or missing. Deoxyribose, as you can see on the 2nd carbon, is missing that oxygen. So deoxy, no oxygen ribose sugar, is the sugar present in DNA. And that tiny difference, 1 oxygen atom, is a clear, and it looks like a boat and a chair. That's how different it is to our cells. Totally different molecules. One tiny oxygen, present or not present, completely different molecule. And our cells don't make mistakes. It's really amazing that such a subtle difference is so clearly different to our cells. And then if you look at the very bottom, we have 6 carbon sugars, hexoses, hex for 6. We have alpha mannose, alpha galactose, and fructose. C6H12O6, same chemical formula. And yet these are very, very different sugars. So if we look at fructose, you can see it forms a little pentamer, not a hexagon, has 5 sides. And so you can see it looks very different from something like a 6-ring sugar. So we have the true hexoses, which are that hexagon shape of the 6 rings, or the 6 sides. And if we look at alpha galactose and alpha mannose, okay, and you can see each carbon is numbered. We have little red numbers. We have a prime carbon, second carbon, 3' carbon, 4' carbon, 5' carbon, 6' carbon. And that's how we refer to them. So you can always say the first prime carbon has the hydroxyl group down. The second prime carbon has the hydroxyl group down, alpha galactose. The 3' carbon has the hydroxyl group up for alpha galactose. The 4' carbon has the hydroxyl group up for alpha galactose. Okay, so see my nomenclature there? So when you're comparing alpha mannose to alpha galactose, I've used this slide for more than 10 years. And if you asked me to draw alpha mannose and alpha galactose today, I could not do it. I mean, I could draw that it's a 6 carbon sugar. I could draw that they're the hexagon. I could draw that. But which hydroxyl group up or down, I would have no clue. I can't tell the difference off the top of my head. For cells, it is a fundamental difference. So alpha mannose sugars are often on bacteria, and we mount a strong immune reaction to mannose. So if you injected mannose sugars into a person, you would stimulate an immune response immediately, because mannose to our cells indicates infection, indicates bacteria. Galactose, they don't care. It's just a sugar we can use for energy. Totally different. So for me, looking at it, every single year, okay, prime carbon, both hydroxyl groups are down. 2' carbon in galactose, hydroxyl is down, but in mannose, hydroxyl is up. 3' carbons, exactly the same. 4' carbon, hydroxyl is down in mannose and up in galactose. So that simple change of just 2 little hydroxyl groups switching between one and the other, completely fundamental different reactions. Immune reaction to mannose, no reaction to galactose. Very, very, very different structures to our cells with very, very, very different functions. One indicates infection and response. The other one, yeah, we don't care. It's a sugar. So structure determines function in molecular biology and in microbiology. So as you can see, when you're talking about organic chemistry, it's often focused on the sugars. And the sugars can take a wide variety of shapes because that H12O6 can form hydroxyl up, hydroxyl down, hydroxyl up, hydroxyl down on all of those carbons. And so that's when you get into really incredibly specific structures. And so as you go from a straight chain form of a sugar and that double covalent bond oxygen, the aldehyde group, can go up or down. As you can see, when you have a C6H12O6 straight chain sugar, can go into alpha D-glucose, in which the hydroxyl is down on the first prime carbon, or beta D-glucose, in which the hydroxyl is up on that first carbon. Now for this class, you don't have to ever be able to tell alpha and beta D-glucose apart or anything like that, but just the concept of how fundamentally different a tiny hydroxyl up or a tiny hydroxyl down is. That's the concept, is cells will use one, but not the other. It's incredibly, incredibly different for cells. I couldn't tell you today which is alpha and which is beta, and yet for a cell, absolutely will use that, but not the other. Totally different molecules, even those tiny subtle differences. So when we're talking about cellulose, that's a straight chain of glucose monomers linked together. And so cellulose is going to be glucose linked together with something called a beta 1,4 glycosidic linkage. Now, most animals, be they termites or koalas or geese or cows, cannot break that beta 1,4 glycosidic linkage and instead rely on bacteria to have the enzymes called cellulases to break open the cellulose made by plants. And so cellulose is an unbranched chain of glucose that fills a structural role in plants. And so linear strands, a single strand of glucose linked together is cellulose. You bundle lots of those cellulose single strands into lots of cellulose together, and then you bundle even more of those cellulose single strands together and you cross-link them like so as you're weaving them together, and you get a plant cell wall. Now, plants can also make starch, which is still glucose linked together, just pure glucose, but they'll be branching. And so both starch for plant energy storage and glycogen, animal energy storage, are polymers, lots of glucose linked together. But for both starch and glycogen, instead of a beta 1,4 glycosidic linkage, it's an alpha 1,4 glycosidic linkage, or an alpha 1,6 glycosidic linkage to make the branches. And so as you can see, starch is partially branched polymers of sugar, of glucose, and glycogen is lots or highly branched polymers of glucose. And animals can break that alpha 1,4 or alpha 1,6 glycosidic linkage. We can do it on our own. We have those enzymes. We do not need bacteria to break it down. So energy storage for plants is starch, energy storage for animals is glycogen, and simply it's glucose, but with different bonds. So animals cannot directly digest glucose with the beta 1,4 glycosidic linkage, making it cellulose. We can't break that down. But if you have glucose as an alpha 1,6 linkage, we can break it down. So it's all glucose, but how the glucose is joined together totally determines its function. Straight chains, cellulose. We can't digest it, and it serves as building material for plants. But if you have branched forms, the alpha linkage, we can break it down. Plants can use it for energy storage. We can use it for energy. We can store it as glycogen. So again, structure determines function. Very, very subtle structural differences, very, very different functions. Straight chain bonds, building material. Branched bonds, energy storage. It's all glucose. So how we define sugars is usually based on the number of carbon atoms. And then how you call each and every single sugar is based on the orientation of hydroxyl and hydrogens at each and every single carbon. So each and every single 5-carbon sugar will have variations on hydroxyl up, hydroxyl down at every single carbon. And so you have 5-carbon sugars, ribose and deoxyribose. Really the only difference between them is on the second carbon, oxygen on ribose, and then D, remember DE means no, oxy, no oxygen on that second carbon for deoxyribose. Now, bacteria use sugars for all different structures. And one important kind of bacteria are something called gram-negative bacteria, which we'll get in during the semester. And so there's something on the surface of gram-negative bacteria called lipopolysaccharide, LIPO for the lipid or hydrophobic component that makes up the lipid bilayer. Poly for a lot and saccharide for sugar is the structure that sticks outside of the cell. And so gram-negative bacteria are like a really fluffy dog or a cat. They're covered with lipopolysaccharide that sticks out. And so we can use that polysaccharide part, which is simply a lot of sugar molecules sticking out of that bacteria. We recognize it with our immune systems. Just that structure that you see down at the bottom can trigger sepsis or septic shock in a patient. I mean, they can have this huge effect on our immune system. And the core region, the part that's closest to the bacteria's surface, is going to be species specific. So we can go, oh, that's E. coli, or oh, that's salmonella, or oh, that is cholera, or oh, that is a bioluminescent bacteria, just based on that core region. And then that outer region will be different between, oh, that is the E. coli that is normal to your gut, or that is the E. coli that will cause hemorrhagic fever. I mean, that's the variability between the sugars. And so we can use that to make vaccines. So there's Streptococcus pneumoniae, and we can actually use some of its sugars on its surface to make a vaccine. The Neisseria meningitidis, it's one that has this polysaccharide on the surface, and we use that to make the meningitis vaccine to Neisseria meningitidis. You have Haemophilus influenzae, also has sugars on its surface. You use those sugars to make a vaccine, and you have protection. So you can use sugars for structure. You can use sugars for energy storage. You can use sugars for your outside structure. You can use sugars for vaccines. So they have a huge variety of functions. Okay, our next macromolecule that we're going to talk about are lipids. So lipids make up cell membranes. Now, for most living things, such as all eukaryotic cells and all bacteria, these tend to be lipid bilayers. We'll talk about one special group of prokaryotic cells, the archaea, which kind of break this general rule. Some of them have lipid bilayers, but some of them have something called lipid monolayers. But all cell membranes have lipids because you need that hydrophobic region to separate outside the cell from inside the cell. Now, eukaryotic cells are often very complex and will have multiple organelles, such as the nucleus, multiple mitochondria, a smooth and rough endoplasmic reticulum, a Golgi apparatus, various lysosomes, etc., etc. Now, prokaryotic cells can also be fairly complex inside, but prokaryotic cells do not have organelles. Lipids can also be used for energy storage. And the basic units, the building blocks of lipids, are going to be fatty acids or hydrocarbon chains, and then glycerol. Now, hydrocarbon chains come in two basic forms: saturated hydrocarbon chains and unsaturated hydrocarbon chains. So if you are saturated, you have a full set of hydrogens, and that every single carbon in a hydrocarbon chain will be covalently bound to two other carbons and to two hydrogens, as you can see. So if you look at A, the palmitic acid, all the central carbons go covalent bond to carbon, covalent bonds to two hydrogens, covalent bond to carbon. Now, the carbon atoms at the end, of course, have to have three covalent bonds to hydrogens, but because carbon believes sharing is caring, that is going to be a hydrophobic hydrocarbon chain. And so CH2 equals sharing of electrons between the carbons and the hydrogens. Now, if you have unsaturated fatty acids, that means there's a double covalent bond in there. Maybe one, maybe two, maybe more. So if you have double covalent bonds, that means you have carbons that have two covalent bonds between another carbon and thus have only one hydrogen attached. Now, as you can see, saturated fatty acids are straight in that the hydrogens facing out forces those molecules to be a straight hydrocarbon chain down. But when you have unsaturated fatty acids, what you get are bends or kinks to it because you have cis or trans interactions with those hydrogens. So as you can see, double bonds between two carbons make an unsaturated fatty acid. The carbon has kinks. So if you are a saturated fatty acid, you're straight and you pack in very tightly. And so saturated things are like butter. Butter is solid at room temperature. It can pack in very tightly. And unsaturated fatty acids, because they have kinks, can't pack in very tightly, and that's olive oil. So saturated fatty acids, butter is solid at room temperature. Unsaturated fatty acids like olive oil, because of the kinks, are liquid at room temperature. Now, if you have a membrane, you don't want a membrane that is butter, that's too tough, too solid, not moves enough. And you do not want a membrane that is olive oil, that is way too liquidy. So most membranes are made up of a mix of both saturated and unsaturated fatty acids so that there is fluidity, cells can move, they can change shape, and there is rigidity, that they have structure. And so you need a mix of both saturated and unsaturated fatty acids. And so here you see a typical phospholipid that makes up a cell membrane, and that you have an unsaturated fatty acid that will have a kink to it, as you can see, due to a cis double bond, same side. And then you have a saturated fatty acid making that nice straight chain. And so by mixing these together, you can have a cell membrane that can move and take stress and take tension, and that has rigidity so it can support itself. And so these hydrophilic heads will face out into the outside environment and into the cytoplasm or intracellular environment. And the hydrophobic tails will face each other, separating outside the cell from inside the cell. And so these hydrophilic heads and hydrophobic tails naturally align. Weak forces always want to maximize each other, so hydrophilic heads will always face out or in, and hydrophobic tails will always face each other. And so they will spontaneously assemble like this. And that allows us to make cell membranes. And so because you have the aqueous environment outside the cell shown in orange, and the aqueous environment inside the cell shown in blue, you can have separation. Outside the cell is now different from inside the cell. And because you have hydrophilic heads facing out and in, and hydrophobic tails facing the middle, you don't want to pass something that's hydrophilic between the hydrophobic tails. So you have complete separation. Non-polar things stay in the non-polar area, and polar things stay in the polar area. So you can anchor various molecules. So you can have the brown molecule, for instance, is a protein. You have a hydrophilic part of the protein facing inside the cell, then a hydrophobic part of the protein, the coil, trapped in the hydrophobic hydrocarbon tail group, and then another little hydrophilic part. And you can pull on that protein and it won't move. The hydrophilic wants to stay with the hydrophilic, and the hydrophobic wants to stay with the hydrophobic. And so cells use this fact to anchor all kinds of various molecules in their membrane. And so you can see proteins in brown there. All those coiled regions are hydrophobic and will be permanently anchored in the hydrophobic part of the membrane. So you can't move them. They're anchored there. And so if you need a series of proteins to do a job, just anchor them one after the other in that phospholipid bilayer. They're not going to move, and you have them all together. Maybe you want to join to another cell. We'll smack two proteins together with their hydrophobic parts anchored in the hydrophobic area of the phospholipid membrane, and then you join those cells together. And it works really well. And so all membranes kind of look very similar. The red and blue and yellow membrane up at the top is a typical animal cell membrane. You can see the plant cell wall in yellow and green down at the very bottom. That's the membrane of the plant. And then you can see how much cellulose is actually present in every single plant cell. And so you can see the fibers of pectin and cellulose making up the plant cell wall. And then down at the bottom, we have two examples of bacteria cell walls. We have a gram-positive cell wall with something called the plasma membrane and then the peptidoglycan layer. And then we have a gram-negative cell wall with a plasma membrane and outer membrane. And then you can see the hairs of the lipopolysaccharides sticking out. And yet we have phospholipid bilayers, we have proteins anchored in those phospholipid bilayers. And they're all relatively smooth and easy to see, and you see scattered proteins. Now, those proteins can do all different kinds of jobs. If you're a cell, you want to be able to take things inside the cell or maybe put things outside the cell. So a protein will do that. A protein will allow you to take a molecule from outside the cell and move it across the cell membrane, or take something from inside the cell and chuck it out. Enzymes will be anchored in the plasma membrane because you maybe want to make one thing into a final product. And so you take in this example, the triangle, one enzyme converts it to the square and another enzyme converts it into the sphere. You need all those enzymes together, put them in the cell membrane. They're always going to be next to each other. It's easy to do that catalysis interaction. Other jobs proteins do in cell membranes is receive signals. Insulin is a protein we make. We circulate through our body. Our cells receive the insulin through a protein receptor in the plasma membrane. Cell recognition. When we have our immune system checking for infection, we use proteins to do that. The reason we don't rip our intestines apart are proteins linking intestinal cells together so that when smooth muscles squeeze them, we have tight joints between all of those cells. All of our cells are joined together so we don't just fall apart. That's all through proteins anchored in phospholipid bilayers. Attachment to the extracellular cytoskeleton, intracellular cytoskeleton, all those connections are all done through proteins. To get molecules into the cell, very few things can diffuse across the cell membrane. And this is typically our steroid hormones and not much else. Everything else really takes a protein to get it across. Now you can use proteins to do facilitated diffusion, in which a molecule, here the example of triangles or spheres, will go from high concentration to low concentration using a protein partner to move across the cell membrane. That does not take energy. But if you want to move something from from low concentration to high concentration, that is much harder and takes energy. And that would be active transport, moving something that is already at a low concentration to a high concentration. That takes energy. Now, in all of these pictures, you see just the occasional little proteins sitting in the plasma membrane, like over here, they're just proteins scattered about. And like for the upper example of the red, yellow, and blue, there's little green dots, and those are actually polysaccharides. And it looks very plain and scattered and distant. And that is simply for clarity's sake, because if we actually investigate the surface of cells, actually look at that phospholipid bilayer, this is kind of what it looks like. So this is a red blood cell. Red blood cells are the simplest cell in our body. They have no nucleus. They have very, very few proteins. They have a lot of hemoglobin, but they have very few other proteins. They're essentially a plasma membrane around hemoglobin, 97% by dry weight hemoglobin. So there's almost nothing in them. They don't have to do any job other than bind oxygen and move it around the body. And so it has the simplest membrane of any cell in our body. And this is what it looks like. The cell membrane, as you can see in pink, is just absolutely covered with proteins and sugars. You can see the proteins in blue, kind of the column things sticking in the membrane. All of the yellows and oranges, that is all sugar molecules sticking out, going, hey, I am this red blood cell for this particular person. I am the O sugar or B sugar or A sugar or AB blood. And all of that is identification. And this is the simplest, smoothest cell membrane in our body. Okay, and that's what it looks like. And so if you look down at this bottom corner, you can see RBC, the red blood cell. So it has to squeeze through our capillaries, squeeze through our arteries, squeeze through our veins. It is the smoothest surface of any cell. And then we have next to it a typical endothelial cell. So the cells that make up our capillaries, the cells that make up our arteries, the cells that make up our veins. That is the typical structure. So that is a smooth tunnel for our red blood cells to go through. And you can see how absolutely massive most cell structures are on the plasma membrane. They are just covered with molecules, just squeezed and packed in, hundreds of proteins, hundreds of sugar molecules sticking into that plasma membrane. So they are very, very complex structures. Okay, that is it for the first lecture. Thank you all. So I will see you all for the next video.